And we have 16 people so far. Okay. So can you see my screen?
Yes.
Okay, great. Okay, so hi everyone. Today we're gonna talk about NetPyNE and how to use this tool to build multiscale models of brain circuits. My name is Salvador Dura-Bernal. I'm an assistant professor at the State University of New York and a principal investigator in the grant that funds NetPyNE. So today I'm gonna start giving an overview of what the tool is and how it got started and how it can be useful for you. And then we'll go into a 6-hour-long tutorial with many different components. So here's the schedule for the day. So the first part is this 30-minute introduction to NetPyNE. And then we'll have a session where we'll go over the graphical web application. So NetPyNE is a tool that has both a graphical tool that's online, as well as a programmatic version where you can, using Python, code the same things you do graphically. And so the first session is going to be using the graphical tool. Then we'll have a 30-minute break. And in the afternoon or in the second session, we'll do the programmatic tutorial. I'm also planning to have some breaks, 10-minute, at least what 10-minute break in each of the sessions, since it's over 2 hours. And yeah, please interrupt me anytime. So this is supposed to be like a hands-on interactive tutorial. So the idea is that everyone can participate at any time, ask questions, and feel comfortable to interact with everybody else. And okay, so I'll start giving this introduction to the tool. So NetPyNE stands for Networks Using Python and Neuron. It's basically a package, a Python package and a graphical interface that helps build models, develop models, run parallel simulations, and optimize the parameters and analyze the output of these simulations. And basically, we're building simulations of the brain and neural circuits. The underlying tool that we use to run these simulations, the backend simulator, is called Neuron, which is one of the most widely used tools in the field. And the NetPyNE tool has been funded by NIH over the last 3 years, and there's 2 years remaining of funding. Hopefully, we also get some additional funding in the future. So the motivation to develop NetPyNE started when I was building these very detailed models of the motor cortex circuits, working with another experimentalist called Gordon Shepherd. And he provided tons of experimental data at different scales. So we had information about the morphology of the neurons, the biophysics of the neurons. We had data about the connectivity, how the different cell types in this circuit connect. We had information about how long-range inputs from other parts of the brain connect to these cells. And so it was pretty hard to start building these very complicated models at different scales using the available tools. So that was one of the reasons we started building this high-level tool for brain circuit modeling. The other big one was that we want to separate the low-level code from the actual biological parameters. So right now, for example, if you go to ModelDB, which is this database of models in Neuron, you will find hundreds of different models that have been implemented, but they are typically quite hard to understand, and particularly to find out what are the biological parameters that were used to build the model. So in NetPyNE, we wanted to separate these parameters from the underlying implementation and using a standardized format. So this makes it very easy to read any models that exist already or that are being developed, to interpret what they mean, the different components and parameters of the model, modify the model, share it with other people, and so on. So here in the left, you see an example of how cells are defined in NetPyNE with this sort of quite transparent structure. We have a pyramidal cell in a location of 100 to 400 micrometers, 50 cells. And then what would be the typical way to implement these models in Neuron, where you have a lot of code, low-level Python code that makes it harder to understand. The third motivation was to facilitate parallelization of the models. So as soon as you start building these large-scale complicated models, you require this supercomputing or at least parallelization on your local machines. And although this is supported by Neuron, it is not always easy to set up. So for example, you have to divide the cells among the different nodes so that each node of the supercomputer, for example, simulates different cells. At the end of the simulation, you have to gather back all the data generated at different nodes and combine it together. So in NetPyNE, we make this automated and transparent, so the user doesn't really have to do anything. And we also facilitate what's called parameter exploration or optimization, where you basically want to run your simulation and test different parameter values. So you might change, I don't know, the synaptic conductance of a particular synapse and see how that affects the simulation. And so NetPyNE makes it very easy to set up these parameter explorations in an easy automated way. So to clarify the relation between NetPyNE and Neuron, here you have at the base of the base level, it's Neuron, where NetPyNE builds on top of this. So in Neuron, if you wanted, for example, to create a synapse, this is the code that you would have to use. And you see it's quite complicated in a way. You have to know several components and run several instructions. You have to create a synapse, then you have to create a connection object and set the parameters. And this is for a single synapse. Whereas in NetPyNE builds on top of Neuron, and you can define connections using these connectivity rules, which are human readable and allow you to create thousands of connections at the same time with a single rule, where you specify the conditions of the presynaptic populations, the conditions of the postsynaptic populations, and some properties of this connection rule, a probability of connection, for example. And this in itself can create thousands of connections at a time. And finally, we have the graphical interface or graphical web application, which essentially allows you to do the same that you can do programmatically via these Python scripts, but using a graphical interface. So it's quite helpful for people who are starting, for example, who just want to play around and see what NetPyNE can do. Here's an overview of the different components in NetPyNE. I'm not going to go into detail here, but just to highlight that we have sort of different steps in the process of building the models. And the main one that the users will be involved in is these high-level specifications here, where you basically define what you want to include in the model, from the cell properties, the connectivity, stimulation, and so on. And then NetPyNE takes that information and builds the network for you, allows you to run parallel simulations, and then to analyze and save and export your data. In terms of these high-level specifications that I mentioned, this is where the declarative language or standardized language that we mentioned comes in. So declarative means that you basically tell NetPyNE what you want to obtain instead of how you want to create it. So it's a more easy to use language. And here you can define all the components of a circuit model, a brain circuit model, including the populations, the different cell types in your model, how are they distributed spatially, the properties of these different cells from morphology, biophysics, including molecular processes, which we will see in a bit. Also synapses, different types of stimulation to your network, and of course the connectivity rules, how all these different cells are connected together. Finally, we also have what we call the simulation configuration, where you specify things like the duration of your simulation, what you want to save and analyze, how to visualize your output, and so on. An important component of NetPyNE is that it supports molecular reaction diffusion. So this is a component that's part of Neuron. It's called RxD. and it allows you to define intracellular and extracellular ions and proteins, so different species they're called, such as calcium, potassium, IP3, as well as different organelles, so structures within the cell, such as the endoplasmic reticulum or mitochondria. And then you can define interactions between these ions and proteins and the organelles, for example. You can model diffusion, you can model different processes like phosphorylation, buffering, second messenger cascades, and so on, which of course all have important implications for the functioning and the response, the electrical response of the neuron. So in this way, we can couple the behavior of these molecular level processes to the physiological response of the neuron and in turn of the network. So for example, these molecular processes could affect the firing rate of the cells or plasticity in the cells, which of course are very relevant to the types of model that we want to build. Next, of course, we've spent a lot of time thinking about connectivity, which is essential for circuits. So we have very flexible connectivity rules that can depend, for example, on the presynaptic and postsynaptic cell properties, like the cell type or the location of the cell. We have different connectivity functions available, can be probabilistic or based on convergence or divergence. You can also provide a custom connectivity rule or connectivity matrix that you obtain from experimental data, for example. And different parameters of the connectivity can depend as well on these spatial properties of cells. So for example, you can specify connectivity delays or probabilities that depend on the distance between cells or even the cortical depth of a cell. Finally, we also have built in some learning mechanisms like STDP and reinforcement learning, as well as gap junctions. And here's just an example of how these high-level specifications look. So we have this simple format using Python lists and dictionaries, which is quite intuitive and human readable. And as you can see, everything you have here are just the biological parameters, and that's what you have to provide to NetPyNE, and you don't have to worry about these low-level programming and coding. As mentioned, then NetPyNE will take these high-level specifications and create a neuron network for you. So this is the sort of hierarchical structure of the network, where you have the populations of cells at the top here, and then you can navigate hierarchically within this Python structure and access all the different cells in the network, the properties of different cells, the mechanisms, different ionic channels, for example, within the cell, synapses, connectivity. Everything is then accessible for you, so you can actually modify the neural network as you wish afterwards, so you can customize it also. And you also have access to all the neuron objects themselves. Basically, you can do anything you can do with neuron, you can also do with NetPyNE after it's been instantiated. As mentioned, NetPyNE takes care of the whole parallel simulation process I mentioned before. And you can do these parameter explorations in an automated way. It's also been customized so that you can actually run on supercomputers with different different setups like SLARM or PBS torque, and you can just change a very simple configuration file and run in different environments. We have different types of parameter exploration or optimization methods, also including evolutionary algorithms that are built into NetPyNE, and for example, can try to tune the behavior of a network from non-realistic to physiological responses automatically using this evolutionary algorithm that you can set up pretty easily. And of course, NetPyNE also provides built-in analysis and plotting for the most common plots in neuroscience, from connectivity measures like this connectivity matrix to recording and plotting of the different currents and voltages within the cell. Here you see the soma membrane voltage, raster plots, spiking raster plots with the spike times of the different cells. You have statistics. You also have information measures that you can compute for the network. And we've also included the ability to record, to simulate and record and visualize local field potentials. So this is quite a useful feature because many experimental papers have data on local field potentials or current source density, which we'll see now. So this allows you to compare the simulation outputs to experimental data. And NetPyNE allows you to place LFP electrodes arbitrarily within the 3D volume of your network. You can place them at any location, and then NetPyNE will record the signal, taking into account the currents of all the neurons in your network. As mentioned, we also calculate now current source density, which is derived from the local field potential signal and gives you additional information of the sinks and sources of the currents in the network. And recently, we have interfaced with the LFPy kit package, and this allows us to also calculate what's called the current dipole moments from the different cells, and in turn, the EEG signal, electroencephalogram signal. In this case, we're using what's called a human head model. So we're placing electrodes, the EEG electrodes, in a simulated volume that simulates a human head. We're working on getting also volumes that simulate other species like mice and macaques. But yeah, this is useful again because you can compare the output of your simulations to all these EEG measures that are available in the experimental literature. And of course, you can have measures, as we can see, at all scales, from voltage traces to LFP to EEG. Finally, just wanted to mention here that we are also working with another group to simulate the bolt signal. So that this is the signal that is used to calculate fMRI signals, and it involves a very detailed neurovascular coupling model where you take into account other aspects of the neural simulation, like astrocytes and the blood level, the blood flow. And so at some point, we should be able to generate equivalent fMRI signals as well. As I mentioned, NetPyNE has a graphical interface, graphical web application that we have been developing for many years now. And the idea is that this would help people who are not so familiar with coding and programming, including maybe clinicians and experimentalists, to use our tool as well. So this web application allows you to do everything that you can do with NetPyNE, basically, from creating the models to plotting and running simulations and plotting the results. We are going to be using this web application for the first half of the tutorial today. And here's an example of how you define the different components. I won't go into detail because we're going to see a lot of this today, and how you can then visualize the output of your simulations. Here is a few pyramidal neurons and the voltage traces of these neurons.
I've got a question relating to scope.
Yes.
If you don't mind me jumping in.
Of course.
So from our work, we do a lot of calcium imaging and increasing amount of optogenetics. So I was wondering whether there are any specifics. Obviously, you can plot out the calcium dynamics within the neuron with the existing configuration, but have you got any plans for implementing, explicitly implementing things like GCAM or channel rhodopsins, for example, for optogenetics?
Yes, no, great question. And we've really thought about this recently because a lot of people are demanding it. We haven't got anything explicit, but as you can say, of course, you can sort of customize your code and plot these things as well. There's a couple of students that have been working on optogenetics, simulating optogenetics. But I think it's a great idea to sort of provide a built-in version of this, both calcium imaging and optogenetics, that you can play around directly, for example, from the graphical interface.
Okay.
Thank you. And yeah, so this graphical interface, this is another example of some cells that are visualized here. And one very exciting extension to this interface that we've been developing recently is the ability to perform these parameter explorations that I was mentioning before. So you can now set up within the web interface the parameters that you want to explore. For example, here we are exploring the weight of connectivity. We can provide a list of values that we want to look into. And then via the web interface, you can run all the simulations automatically and look at the results. So here is on the left-hand side, you see the results of this parameter exploration, and you can click on each of these ones and visualize directly the results of these simulations. So this is, I think, quite useful because as soon as you start building models, the first thing you need is to explore parameters to perform any actual useful research. So we're very happy that you can do this from the graphical interface now. And of course, our graphical interface is fully integrated now with open source Brain. So you can see here at the top, the open source Brain platform. And this has been a great advancement because open source Brain allows you to have your own workspaces, permanent workspaces. So you can have your own file system that is permanent. You can come back again day after day and keep working on your own environment. You can create different workspaces with different files. You can clone, for example, from GitHub repositories, also from built-in repositories on open source Brain. And of course, then you can use some of the other tools that are available in open source Brain, such as the NWB Explorer, which allows you to visualize experimental data directly from within your open source Brain environment. So today we're going to be running all the simulations on open source Brain. And yeah, you'll see the great benefits. We're also playing around with another tool that's available on open source Brain, which is the Jupyter Lab. And that will allow you to also run using Jupyter notebooks the same simulations and models that you're running using the graphical interface. So yeah, we'll see a lot of that today. We've also integrated with another platform called the eBrains platform, which is part of the Human Brain Project, and you can also run NetPyNE models there. And we are working on another exciting aspect, which is an interface with the virtual brain tool. So the virtual brain essentially allows you to simulate whole brain models. So you can simulate the activity of the whole brain, which is divided into a number of nodes, for example, 50 nodes for different regions of the brain. And so what we've done is allow you to select one of those nodes and then replace it with a detailed model built in NetPyNE. So the rest of the brain will be still simulated in a less realistic or detailed way. And then one of the nodes will be built using a very biophysically detailed model. And both of those will speak to each other. So basically for us at the NetPyNE level, it means that we will have very realistic activity of the whole brain that actually gets modified with the activity of our own model. And we can then explore the relationship between the local processing at the microcircuit level and the whole brain activity. Both simulators will run at the same time. It's with something called co-simulation, and they will exchange information, essentially the spike times and the average firing rates of different populations, and yeah, basically interact with each other in real time. This is just an example of this interaction happening. And just to finish off this part, I just wanted to mention we have a lot of documentation, tutorials, and examples available online. Here are some of the links. We have documentation, we have a package index with all the list of different functions available. We have tutorials, we have Jupyter Notebook tutorials as well. And we have videos on YouTube, over 40 hours of videos showing how to use both the graphical web interface and also the programmatic mode of working. We also have a list of example models that have been developed by people or are being developed right now. And you can click here and basically navigate through all the existing models with links to different repositories. And we have a Q&A forum where people can post questions. There's a lot of them already there, so it's a good resource if you have any issues. We have a publication on eLife from a couple of years ago if you want to know more. And what we're very excited about is the sort of the growing community that we've seen and the number of models being developed right now. So there are over 80 models that we know of, developed in over 40 different labs around the world. Here's an example, a list of some of these example models that I've put together. And as you can see, we have on the one side, models of different disease and disorders from schizophrenia, ischemia, epilepsy, chronic pain, depression, Parkinson's. We also have simulations or models of different treatments, TMS, optogenetics, which was mentioned before, ketamine, and different measures that I've already mentioned, EEG and fMRI. And we have models of specific regions of the brain, lots of different cortical regions, motor, somatosensory, auditory, and visual. We have this very famous claustrum region, cerebellum, spinal cord, prefrontal cortex, and even more subthalamic structures like the amygdala, hippocampus. Well, hippocampus is in the cortex. And we even have models of cardiac circuits, which are not really part of the brain, and of the gastrointestinal circuits. So all of these are in the list that I mentioned before, and you can have a look at them if you're interested. And here is a list of publications that have used NetPyNE already to build these models. So there's around 20 of them already, and we know of several that are in preparation right now. And this is just an example of what came out recently, which is a model of schizophrenia developed by Christoph Metzner and Volker, which was published recently in Nature Schizophrenia. Okay, so I had a couple of quick examples here of existing models in NetPyNE, which I'm gonna go over them pretty quickly because it's already 9:30. But one of the models that we've been developing for a while and it's now under review is this model of motor cortex, which includes 10,000 biophysically detailed and morphologically realistic neurons with 30 million synaptic connections. You can see here a bit better the structure. So all these different cell types are all tuned to real physiological data and to morphologies. And the connectivity is also based on many papers from the same lab, from this Gordon Shepherd's lab, at the local microcircuit and long-range connections. So what we've done more recently is, yes, full screen, is validate this model. So this is just showing the connectivity that we mentioned based on experimental data. So we've validated this model. Oh, sorry. And yeah, just to illustrate the computational capacity that's required to run these models. It takes around 2 hours on 100 cores to run 1 second of simulation of this model. But we've recently been able to run it with Core Neuron, which is this optimized engine by the neuron developers. And we've seen speedups of up to 40 times speedups, which we are quite excited about. So yeah, if you're interested in running these large-scale models, please look into Core Neuron, because it now allows also not only on supercomputers, but also on GPUs to accelerate your models. And as I was saying, what we've done with this model is, of course, the first step is always validating the model against data. And in this case, we've used in vivo data from Ian Dugit's lab in Edinburgh, who has a very interesting setup where the mice are running, performing a task, and they record in vivo from these different cell types, whole cell recording. And so we validated the different cell types in the model just to show that the model reproduces the activity and their different behaviors of these mice. We also validated the local field potentials. So this is for different behaviors, quiet and movement. This is the experiment and the model. So statistically, we show that we see the same sort of features and changes in features in LFP power. So there's, for example, a decrease in the delta oscillation power and increase in gamma. And yeah, I didn't mention here, but of course, we've used the model to then make predictions about motor cortex and as well as motor cortex disease. The other similar model that we've been building is of macaque auditory thalamocortical circuits. And here, I just want to illustrate this because we've done measures across all these scales, and this really illustrates the multi-scale aspect that we highlight in this course. So from this model, we can obtain from membrane voltages of individual cells to the spiking activity of the network, the local field potentials, and now these EEG signals as well. We've also validated this model against experimental data in vivo. And of course, the model allows you to make predictions and try to explain where these different oscillations come from, for example. The model allows you to break apart the contributions of different cell types to the different oscillations that, that you can measure in the simulation. And finally, just quickly mentioning this S1 model, which is comes from the famous Markram 2015 paper, where they built this very detailed S1 model with 30,000 neurons, 40 million synapses, and many different cell types and pathways. We have now converted that model into NetPyNE, and it's also currently under review. And we've validated so to make sure that the cell types reproduce the same behavior as the original model, and also the connectivity has the same properties. We've extended the model to actually include thalamic circuits. So we have different cell types in the thalamus that are connected now bidirectionally with the S1 somatosensory cortex model. And we can show activity in the full S1 model with all the neurons in the S1 model and all the neurons in the thalamus here at the bottom. You can see this is the new thalamic circuits that have been added. And yeah, we show some interesting dynamics, synchrony between the thalamus and somatosensory cortex and so
on. Salvador?
Yes.
Right. Just a quick question. So some of the graphics you showed there of the reconstructed circuits, these weren't generated by NetPyNE UI, these were generated offline?
These were from the original or from some other version of the S1 model, yes, that we just got to from the website.
Can I also jump in?
Yes.
So I really like the experimental validation of the M1 model. I was just wondering if you'd done anything similar with the somatosensory model in terms of validating against in vivo data rather than, because presumably it's mainly based on the slice work of Markram.
Yes, we haven't done anything beyond what they did in the original paper, but we have the sort of slice version, and we also now simulated for the actual revision of the paper. They asked us to simulate the low calcium version, I think it is, which in theory simulates the more in vivo conditions. And yeah, we were also able to reproduce the same results that they show in that paper, but nothing beyond what they did already.
And another very quick question, sorry, about the data for the M1 model. Is that available? Is that going to be made available, or is that part of the paper?
It's under review. So as soon as it hopefully gets accepted, we will make everything available. Yeah, all the
including the data from Edinburgh.
Yes.
Okay. Is that going to be NWB?
It should. Okay. I don't think that it is right now, but we can convert it.
Okay. No, that's fine. Thanks.
Yeah, so that's all for now. So just a quick shout out to everybody who was involved in this work, as well as the funding from NIH and NSF. And yes, if there are any questions, any other questions?
So do you envisage any issues in terms of running some of these very large models with this core neuron advance through something like OSB? I guess there's a issue of getting the data back as well, because depending on what you record during your simulations.
Yes. I think something similar has been done for this NSG platform, where you have these interfaces and you send back the data back and forward. I assume if you run these very large scale models, we might have some issues because we already seen them when we actually run them on the supercomputers themselves. It's quite hard to analyze the data in place and also to move around. Particularly for the graphical versions of the interfaces, we cannot really visualize anything larger than 500 neurons or so. It's something that we have in the grant as one of the extensions that we want to work on, to try to visualize larger models and analyze larger models. But of course, yeah, it's one of the issues right now.
Okay, thanks.
Any other questions? Okay, so yes, so here we have the schedule. So we finished this first part, introduction, and now we're going to go into the graphical web application tutorials, which, as I mentioned, are on the open source Brain platform. So the first step, oh, I forgot to mention for everyone, let me just go to the beginning. I think it would be quite useful for you to download the slides, and you can find the link right here at the top. It's one of these short links, bit.ly/NetPyNE online tutorial 2022. You can then access these slides online and follow along, because we've added slides for all the steps of the tutorial, both the graphical and the programmatic one.
Can you put it in the chat? Yeah.
Yes, that's a good idea. So I've added the link to the chat, so you can now download them. Okay, so yeah, so as we go through these tutorials, please, if you have any questions, you're stuck, you fall behind, you forgot one step, let us know through the chat, or just talk. You can unmute yourself and start talking. You can also use the slides, at least for the first two components. There's a very detailed step-by-step slides for what we're doing. So if you get lost, you can also just look at the slides and you will see each of the steps in a different slide. But yeah, feel free to interrupt anytime. So the first thing we're going to do, since we're going to be using open source Brain, is to register to open source Brain if you don't already have an account. And so I'm going to show you how to do that. Okay, so I can open here. I go to v2opensourcebrain.org. And this is what you will see. And you have to go to the top right, sign in. I already have an account, so my information is here. But if you don't, just click on register. And filling the information is very quick, just the basics. And then you can just use that information to log in here. So again, this is also shown in the slides here, so you will see the different steps. And I'll give you a couple minutes just to do the registration and log into Open Source Brain. So can everyone please confirm that you have signed in? Can you please, or maybe the other way around, if anyone has issues, can you please let us know? You can put a message in the chat or just speak up if you have issues registering to Open Source Brain. Okay, so it seems hopefully most people are okay. So I'm gonna continue and just log into my account. Okay, so the next thing we're gonna do is create a workspace. So as I mentioned, these workspaces are where you're gonna have your own files and your own working environment. And in this case, we're gonna create a workspace of the type computational modeling using NetPyNE. So you will see here in the left-hand side, you have different things you can create. And in here, we're gonna create computational modeling with NetPyNE. Just click there and it will request some information. We just basically need to put the name of the workspace. You can choose anything you want. I'm gonna put CNS 22 NetPyNE. Okay, so I'm now gonna create the workspace. And then you should see it here on the left-hand side. So I have a bunch of workspaces already that I've created. And the last one is here on the left-hand side. So what we wanna do is open this workspace using the NetPyNE graphical interface, the website, the web application. So to do that, you have to click on the three dots and select Open with NetPyNE. So again, click on the workspace you just created, three dots, Open with NetPyNE. So when I do that, it will take a few seconds and you should start seeing the NetPyNE web application loading. I think we are 23 now, so there should be enough resources for everyone. Hopefully.
Just to let you know that if usually there isn't as much of a delay loading a workspace, it's just I think there's about 10 people at the same time on the system. We're just testing with larger numbers of users, so usually it's a lot quicker than this.
Yes.
But it should get there.
Again, if you're lost somehow, you can go always to the slides and you'll see here the steps we've done so far, which is just register to open source brain, sign in, and create a new workspace of the type computational modeling with NetPyNE. And then we have opened this workspace using NetPyNE. And what you should see at some point is this screen here, which is the graphical application. Again, as Ulrich mentioned, these delays because we have a lot of people trying to do the same thing at the same time, of course.
Salva, can you try maybe reloading your page?
Yes.
Yeah, mine has loaded already.
Okay, great. And here it is loading now.
How is everybody else doing? Is everybody got it loaded?
You could potentially try a quick refresh of the page. That might help things. Or possibly refresh the page after Salvador's has loaded.
Okay, so here we are. Maybe yet say in the chat if yours is working or not. If you're having issues, let us know how you're doing. Okay, it's working.
All right, looks like it's going.
4, 5. Great. 6. So far we have 6 out of 24. Not too bad.
All right.
And so once you have it working, the first step we usually do just to make it easier to navigate around is just to minimize this left panel, which are the resources, the open source brain resources. So you can click here in the bottom, this arrow, and this will make the NetPyNE interface a bit bigger. And yeah, maybe we'll wait until everyone has it working. But just quickly to mention, so in this interface, what we're seeing now is what we call the sort of building mode, where you're gonna define the model that you want to build. And you see here already, if you expand this column, you see within model specification, all the different components of the model, which I mentioned on my slides, the cell types, populations, synapses, connectivity, stimulation in the network, this reaction diffusion, and then some configuration, settings for different plots, and the overall configuration of the simulation. We also have these tools at the bottom. One of them is the experiment manager, which we're gonna see at the end of the tutorial, where you can create these parameter explorations to run simulations and explore the values of different parameters and the effect they have on the network. And we have what's called the Python console, which allows you to interact with the model from a programmatic way. So you can actually explore the model using Python code. And this is synced in with the actual graphical interface. So anything you change in the interface will be reflected here and vice versa. The same thing is available at the top. So this has the same effect changing on this left panel as changing here on the different panels on the top. And yeah, if anyone is having trouble or still waiting, can they please mention this on the chat so we know? If not, we might just go ahead.
Yeah, sounds good.
So Joe Graham, he's part of the NetPyNE project. He's been developing NetPyNE for a couple of years now, and he's gonna take over and go through the first 2 tutorials, the graphical interface, and then I'll come back to finish it.
Okay, can everybody see my screen here?
Yes.
All right.
It's a bit squashed.
Yeah, try to make it a little bit bigger here. Okay, so again, we're going to minimize the resources panel with that little arrow on the lower left. And yeah, so we got all the tabs and the Python terminal, which Salvador was just talking about. And yeah, so the Python terminal stays in sync with the GUI. So change something in the GUI, it affects the Python, change the Python, it affects the GUI. So yeah, we're just going to build up a very simple network model here using the GUI. So in our NetPyNE GUI here, like Salvador was saying, these tabs on the top and also on the sidebar here, if you prefer, are all the steps you need to go through to build a model of nervous system. So you need to define some cell types, make populations out of those cell types, define the synaptic mechanisms and how different populations or cells connect. You probably want stimulations so you can make things happen. We've got reaction diffusion in the GUI now, which is really exciting. So you can do reaction diffusion setups right in the GUI now, and plot settings configuration. So let's start by, maybe I can zoom in a little bit more even. Yeah, so let's click on, in our cell types tab here, let's click on the plus to create a new cell type. And there's a variety of options here: an empty cell, a basic Hodgkin-Huxley cell, which just has a soma with the Hodgkin-Huxley parameters. Ball and stick Hodgkin-Huxley cell, which has a soma and a dendrite compartment, so it's a 2-compartment model. Or you can also import cell templates from file. In this case, we're just going to use the ball and stick Hodgkin-Huxley cell, just so we don't have to type in all those parameters by hand. So let's click on our ball and stick Hodgkin-Huxley cell, and it pops up our cell type 0 here. But let's go ahead and rename that PYR for pyramidal. So we'll click on our cell type 0 here. And then in the name of the cell type, let's just enter PYR for pyramidal. Great. Now, in the cell, a cell is made of a bunch of sections: a soma section, each little dendrite section. And these sections, in this cell model, we only have 2 compartments, a soma and a dendrite. If we look at the soma, each compartment is going to have geometry and topology. So the geometry of our soma, it's got a diameter and a length of 12 microns. So it's essentially a 12-micron sphere, axial resistance, membrane capacitance. The topology, the soma is not going to have any topology because it's the parent section. It does not connect to anything else. Other things connect to it. But if we look at the dendrite, then we can see in the topology, it's connected to the soma. The soma is its parent, and it connects at the end of the soma. It connects at the beginning of itself. Yeah, so a cell is made up of sections. Each section is going to have mechanisms inside it that allow it to interact electrically, with all the other components of the model. In the dendrite, the only mechanism we have is passive. So it's a passive dendrite. It's just a leak current. In the soma, we should see more mechanisms. So we got the Hodgkin-Huxley mechanism in the soma. And you can have a bunch of different mechanisms in each compartment, as many currents or channels as you need. And yeah, so again, if we were going to build this up from scratch, we could go to section, go to create new section. If we wanted to add another dendrite or an apical dendrite or anything, we would add it here. But yeah, so we've got our cell type. We explored the mechanisms and the geometry. Great. So, normally your model will probably have a bunch of different cell types, but for this one, we're just going to use one cell type, and we're going to make a population out of it. So we're going to click on our questions at any time. If I'm going too fast, please let me know. I can't see the chat or anything, so please somebody tell me verbally if there's any problems. Clicking on our populations tab, we want to add a new population, so we'll click on the plus there. And we're just going to give it the name E for excitatory. You can name it whatever you want. This is just a string, it's arbitrary. For the cell type, we only have one cell type we created, the PYR, so it's going to be that type. And now we want to determine how many of these cells do we want. So we can specify a number. We can also specify a density. So how densely are they in the area? We can specify grid spacing. We're just going to keep it simple and do just 40 cells. In here, you can play with the spatial distribution. So you can set a range of places where this cell can appear. So if you're doing a cortical model, obviously layer 1 cells are above layer 2 cells are above layer 3 cells. You want to set ranges for these things. Great. So we've got cell types and we've got a population. So at this point, we can actually create the network and just take a look at what's available here. So yeah, we got that ball and stick model that we put in. We got 40 of them, and it's just spread out in our default little space there. I think it's a 200 micron square cube. One thing we can do here is on the left here, if we click on the little gear for the control panel, then we can we can add some color to the cells just so they're a little easier to see, to distinguish. Great. So yeah, that's our model in space. If we look over here on the left, these are all the analyses that will be available. None of the lower ones are available because we haven't simulated anything yet. But we have created, it's got no connectivity yet. For our 2D network here, we can see where the cells are in space, but there's no connections between them yet because we haven't specified those yet. So yeah, let's go back to edit and continue building up our model. We've got cell types, we've got a population. So now we're going to need a synaptic mechanism if we want these cells to be able to talk to each other. So we PYR 40 cells, created the network and looked around. So now we want to add a synapse. So let's click plus on our synapse here. And we can name it whatever we want. Let's just call this EXC for excitatory. There's a few built-in synapse types in neuron, exp-syn and exp2-syn. This is just a double exponential, so an exponential rise and an exponential decay. And you can set the time constants for those. So yeah, we're going to set it as exp2-syn, the double exponential, and yeah, a fast rise time of 0.1 and a slower decay time of 1. And a reversal potential of 0. So a fast rise time, 0.1, a slower decay time, and a reversal potential of 0. Okay, so we've got a synapse defined now. Now we have to specify how these cells are going to be connected using this synapse. So we want to click on the connectivity rules tab now. And we're going to want to add a new connectivity rule. So click on the plus. Again, this is just a string, so it's an arbitrary name, but typically it's good to give informative names. So this is going to be connectivity from our E population to our E population. So I'm just going to call it E arrow E, E to E. For the weight of this, okay, we're going to dend postsynaptic 0.5. Okay, give me 1 second here. Where is the Okay, here we go. So we want 0.1 probability of connection. Hold on one sec. I'm jumping ahead of myself here. Okay. So yeah, the weight, we're going to do 0.005. The probability of connection, there's a variety of ways to specify how your cells are going to connect. We're just going to use a very simple probability of connection of 0.1. So every cell has a 10% chance of connecting to any other cell. You can also specify convergence. So how many presynaptic cells come onto any postsynaptic cell. You can do divergence, which is how many postsynaptic cells does each presynaptic cell touch. So there's a variety of ways to do it. For a connection delay, we're just going to put in a delay of 5 milliseconds. In NetPyNE, it's pretty easy to do a variety of connection delays. The most common and useful one is probably a distance-based delay. So the further away two neurons are, the longer the delay between activation of the presynaptic and touching the postsynaptic. The synaptic mechanism, we only have the EXC one that we created, so we're going to set it to that. And for the postsynaptic section, we are going to want it to come in to the dendrite. And now you're going to have to remember the name of your section. If you remember, we called it dend, D-E-N-D. So we're going to put it into there. And let's put it in at the very end of the dendrite. So at 1.0. 0 would be the beginning of the dendrite. 1 is the end of the dendrite. We'll put it in at the end. We're going to have one synaptic connection per synaptic contact per connection. We're not going to do any plasticity right now, but it's fairly easy to put in plasticity rules for your synapses also. Great. So now we need to set the, so that was sort of the general connectivity stuff. Now we need to set, okay, which presynaptic cells does this come from and which postsynaptic cells does it go to? So for the presynaptic, we're just going to go from the entire E population. And again, you can also do it by cell type. So you can connect different types of cell types. You can connect different ranges of locations, but we're just going to connect it population to population, E to E. So in our presynaptic, we just want to set the population to E. In the postsynaptic, it's just going to feed into E. So we're going to set the population to E again. And great. So that should set up our connectivity rules. So we're going to click on update our network again. And now if we click on our 2D plot net, now we can see that our cells are connected. And each one is randomly connected with a 10% probability to any other cell. Okay, so let's go back and finish up our model. So we've got our cells, our populations, our synapses, and our connectivity. Now we're going to want to input a stimulation just to kick the network into gear. Otherwise, it just won't start doing anything. So yeah, we went from E to E in our connectivity, and we were able to see the connections. So yeah, we want to click on the stim sources tab now. And in stim sources, we're going to want to add a new one. And we're going to add an I clamp. So let's just call this I clamp 0. NetPyNE has a neuron has a variety of built-ins that we can use. So we're just going to use the I clamp, the current clamp. We're going to set a delay of 20 milliseconds. That means once our simulation starts, after 20 milliseconds, this I clamp will turn on. We're going to have it turn on for 5 milliseconds before it turns off. And we're going to give it an amplitude of 0.1 nanoamps. Okay, so that is our stimulation source. But now we need to have a target for it. Otherwise, it's not connected to anything. So let's go to our stimulation targets tab. And we're going to click the plus to add a new target. Give it a descriptive name. So this is going to be I clamp 0, and we're going to feed it into cell 0. So I'm just going to call this I clamp 0 to cell 0. Stimulation source is going to be the one we defined. target section is going to be the dendrite. So our current clamp will feed into the dendrite of it, and we'll put the simulation in at the very end of the dendrite. Great, and then we want to click on conditions. So we got the general stimulation stuff. Now the conditions, which cells is it going to go into? You can put it into an entire population or an entire cell type or a range of locations. We're just going to put it into one cell. So we're going to add a new target GID, global ID, and we're going to feed that into our first cell, which is cell 0. Great. Okay, so we've got a stimulation set up. Now we are going to go to our configuration and just make a few little changes. The default simulation runs for 200 milliseconds or 1,000 milliseconds, a full second. We don't really need that much, so we're going to bump it down to 200. And the default time step is 0.025. This is a very simple simulation, simple model, so we're going to increase that to make it run faster. A time step of 0.1 milliseconds. And yeah, so now we're going to have to set up what we want to record from. Neuron and NetPyNE do not record everything that's going on in the model. There's many, many variables. So any variable that you want to save to look at later, you're going to have to specify by going to record here. And we're going to want to record from cell 0. So let's add new cells to record traces. Oops, float numbers are allowed. So we're going to cell 0 and then click plus. And now we want to say which traces from cell 0 we want to record. And we want to, we're going to record 2 traces. We're going to record from the soma and from the dendrite. And yeah, so this is sort of pseudo code you put in to specify the traces. vdend, we want to record in the dend section, and we can record from the very end, and the variable we want to record is V, the voltage. Great, so we should be recording from the soma in the middle, the voltage, and the dendrite at the end, the voltage.
And Joe, just to mention that now by default, it always records from the soma.
That's very convenient. That's, yeah, I just noticed that. That's good. Yeah, so then you can just copy the vSoma and from there and paste it up here, then change it to dend rather than typing it in all manually. Great, so we're going to record 2 traces from that cell 0. Now let's look at our plot settings. Here we go, plot settings. And yeah, so we're gonna, for our traces plot, we want to overlay the soma and the soma and then dendrite voltage traces. We want to overlay those. So we want to overlay the data. We want 1 figure per cell, starting time and ending time. GIDs. Yeah, so then in cells to include, we don't want to include all of them. We're not recording from all of them. So you click in here, go down to GIDs and record from cell 0. So that should do that. Great. One other thing I'm going to change is the connectivity plot on this. So we're going to click on add a new plot. And we want to see the network connectivity plot, all cells. The feature, we're going to do the weight. And group by default, it's grouped by population. So it's the connection between 2 populations. We only have 1 population. So it's a real boring graph unless you group it by cell. And we'll just order it by GID. But that'll make our connectivity plot a little nicer. Great, so now we want to run the or update the network here. So click on the rocket, and then yeah, we're all set. So we're going to click the simulate button here, and that should simulate the network. Great. Yeah. So here's our connections plot. Again, the plots are all on the left over there. And instead of just E to E, one color, this is cell to cell connectivity. So you can see which cells are connected in this connectivity plot. This is the 2D net plot that we saw before. But great. Now we can see more analysis plots. So this is our cell traces. And again, we recorded from cell 0, recorded from the dendrite and the soma. And these are interactive plots. It's kind of cool. So if you click on the legend, you can turn off and on any of your plots. And yes, this is cell 0. You can see the dendrite is the bluer color. It settles into its resting membrane potential until time 20, when our current clamp kicks in at the dendrite. It starts increasing the voltage in the dendrite, which drags up that soma voltage until you hit the spike, the threshold, and you get a spike. And you'll notice in the later spikes, it's the soma that goes, anyway. So that's the traces. The raster plot is a plot of, this is the GUI is pretty cool. You can move figures around and resize them. Let's get rid of these two. Close that. Okay, so now we can see our raster plot and this is cell 0 that we're recording from. So you can see it's the first cell in the network to spike here at, if I overlay it properly, at time 24, it spikes. So the current clamp kicks on at 20 milliseconds, takes about 4 milliseconds to drag the soma up to its spiking potential, and it spikes, and then all the cells it's connected to start spiking. We can see all that in the raster. This is the spike histogram. This is the average cell firing rate over time. You can see it's at 0 at first, and then it kicks on, and you get some oscillations. Granger plot, is that going to work here? Yeah, it does. And this is a causality plot. So all the analyses are on the left here, and you can play around with those. Yeah, so you can compare different plots side by side. Yeah, this is what the connectivity plot would have looked like if it was population E to E. Doesn't do much in the eye clamp. Cell to cell gives us a better, more interesting one. Great. So that was a very simple, building up a very simple network model using the NetPyNE GUI. And at this point, we still need to clear the kernel, right, Solid, before we go on to the next one.
Yes, and Joe, I'm thinking of doing a 10-minute break now, because we are sort of halfway through the session. And then, after the break, we will continue with the tutorial 2, which is a very nice tutorial of morphologically detailed neurons, really beautiful neurons. Then a tutorial of a multiscale network, where we're going to see the effects of changing these reaction diffusion parameters on LFPs and EEGs, a really nice example of these multiscale interactions. And finally, we're going to see how to run parameter explorations directly from the graphical interface. So please come back in 10 minutes. This is a short coffee break. So it's 10:20 a.m. now, US time. So we'll be back at 10:30 in 10 minutes. I'll put the info in the chat as well. Thank you, everyone. If you have any questions in the meantime, let us know through the chat, and we can help you as well. So just to mention to everyone, we are gonna sort of deploy a new version of the interface just with a minor fix that we realized. So your current sessions will close. And so after the break now, we'll have to start a new session for the next tutorials, just so you know. And I'll put this in the chat as well. Okay, so it's 10:30 now, so we'll start back the tutorial. Just in case anyone joined now, this is the CNS satellite tutorial for the NetPyNE tool, and we're halfway through the first session. Joe, yeah, take it away.
Okay, so yes, participants can see my screen. Okay, so yeah, so we redeployed real quick, so we're going to have to restart this, I guess. Let's see here.
I think you can just reload the website.
Oh, just refresh it?
Yeah, refresh it.
Okay, perfect. Refresh the
yeah,
whole site.
So do you want to just mention about the fact that the OSB restarted?
Yes, I mentioned already, also in the chat. So yeah, we restarted to fix a small issue. So your sessions will have finished and you need to restart now, reload the website. And you might also need to sign in again.
All right, loading the workspace here. Yeah, let's see here. Yeah, so now we're just going to do a quick tutorial to show that this works equally well for morphologically detailed neurons as well. So yeah, once you guys get your screen up here, we can minimize that resources tab. And then on our cells tab, we want to create, click the plus to create a new cell type. This time we're going to import a cell template from file. So the bottom option there, after you've added a new cell, is import cell template from file. And yeah, the one we're going to import is this PT cell. And yeah, I think we want to call it PT cell and the cell template class name. Yeah, so the cell rule label, you can name whatever you want. The cell template class name, you actually need to go into the cell model file and see what the name is to call it, to instantiate that cell model. In this file, it's called PT cell, so we'll do that. We want to set the path to the file, so we'll click on our file explorer here. And yeah, so if you upload files, they'll show up in this uploads directory here, but these have already been uploaded for you in, I believe in the tutorials. Yeah, NetPyNE tutorials in the workspace master. And then under cells, we want to pick this ptcell.hoc and select that. If your cell model has mod files associated with it, if you know different channels or things, you're going to need to specify the path to that mod directory, which is again under tutorials in the workspace, and it's this mod directory here. So I'm going to select that. And yeah, we're going to want to compile those mod files so that they're ready for Neuron to use them.
Joe, just to clarify, this is an example cell that we've uploaded previously, but in theory, you can use any existing cell that's built in Neuron. These cells usually come in two flavors, either in this HOC language, which is .hoc files, or in Python, which would be .py, and both of those are accepted by NetPyNE. And so what NetPyNE does is basically extract the information from that cell built in Neuron, and then just store all the values of the cell in the NetPyNE format so that you can use it then in your networks. So potentially you can go to ModelDB, download any file that you want, upload it here to the GUI, and use it directly in the graphical interface for your network.
Which is, yeah, obviously very useful. People have made a lot of great cell models that are out there available, especially on ModelDB, for example. And yeah, you know, when you're building a new model, it's often best to start from an existing model and play around and tweak it rather than starting from scratch. And this allows you to get very complex cell models easily into NetPyNE. Yeah, so that was the PT cell. We're also going to add an inhibitory cell. Oh, well, we can explore it real quick first here. So, okay, here now it appeared. So here's our PT cell under our cell type. And yeah, so this is a morphologically detailed cell. So in the last tutorial, we had a ball and stick model. So it's just a soma compartment and a single dendrite compartment. This model has a soma, an axon, a whole bunch of dendrites, and then also apical dendrites, which grow up like a tree. The basal is like a bush and the apical goes up. And yeah, each one of these is going to have a bunch of mechanisms. So this is what an apical dendrite. It's got some calcium channels, an IH channel, some potassium channels, sodium, passive leak current. So yes, this is a highly complex cell model. So that's an excitatory, a pyramidal one. Let's import another cell template from file, an inhibitory cell that is morphologically detailed. This one I believe we call SRI. And then that's also the class name inside the file, SRI. We're going to click on the File Explorer and go to NetPyNE Tutorials, the workspace, cells. And yeah, so this is sri.hoc file. I'm going to select that. And it's all the mods are in the same, all the mod files are in the same mod directory. So we'll just point to that. They should already be compiled, but we'll click that just in case. And then we'll click import. And that will bring up our SRI cell type here. And then, yeah, we'll make a couple populations and then we can take a look. So yes, this is the SRI, the inhibitory cell model. Again, a bunch of dendritic compartments. Great, let's click on our populations tab and add a new population. We'll call this E for excitatory. And the excitatory cell type will be that PT cell. Yeah, we'll just put 2 cells in. It's a very complex model. It might take a while to load, so you don't want to put too many in. So yeah, we'll just have 2 of the E cells. And then we'll add another population, which we will call I for inhibitory. And the cell type here is that SRI. And again, for the number of cells, we'll just do 2 cells so that, yeah, it doesn't take too long to load. Yeah, so we could add in all the other stuff, but for now, let's just create our network and take a look. And great. Okay, so then we're going to go on the left side here down to the bottom, the control panel, and we can sort of pick a nice color here. Something like that. Close the control panel, and then, yeah, we can zoom in. We can see the pyramidal cells with their apical dendrites stretching upwards, and the more local inhibitory neurons around the base of it there.
So,
yeah.
Joe, if you want to go again to the control panel, and maybe we can explain a bit how this works.
Yeah.
So right now you pressed on the top level, at the network level, and you press the select random color for each population. So what that does is for each population, it picks a single color and colors all the neurons in that population of that color. If you want to have each cell have an individual color, you can go down to each population and select also random color, but at the level of the populations. So at the level of the E, for example, if you go to the right-hand side and select random color, exactly, yeah. Now each of the E neurons will have a random color by itself. And then you can do the same for the eyes. Yes.
Yeah, but if you do it at the population level, it's kind of nice because then all the cells of the same population are the same color.
Yes. So just to explain the different modalities that we have.
And yeah, so at this point we could, well, let's go back to edit. At this point, we could define a synaptic mechanism and some connectivity and stimulation and actually make it happen like we did with tutorial 1. But the whole point of this was really just to show you the morphologies and how that works. If you do want to actually see the synaptic mechanisms in place and see it active, you can load it later on your own from the tutorials tab. The tutorial 1 that we went through, this will load the entire thing automatically. If you click on tutorial 2, it'll load this one along with connectivity and stuff, so you can actually run it.
Actually, Joe, we have some time if you want to show that load tutorial 2 and just show the cells actually spiking.
Yeah, sure. So yeah, so let's click on tutorials and we'll click on 2 detailed cell network. We'll use the old value of net parameters. Do you want to continue? Okay, so we need to restart here before we load it, right? So yeah, on up here on the file, let's go to File, New, and this is going to clear the kernel and everything. So you start fresh. Reload site. Okay, great. And then, yeah, again, in tutorials on the top here, we'll have to go down and click on tutorial 2, the detailed cell network. And that will load that up. Great, here we go. So yeah, let's create, well, yeah, you could have taken a look first, I guess, to see what it did. Okay, let's just give some cool colors here. There we go. And yeah, so let's click on simulate. And again, so with the simple model, it was only 2 compartments. It only has to calculate 2 equations for each one. This has hundreds of compartments, so it's a lot more calculations. It's going to take longer. Okay, so our connections plot, you know, fairly simple. 2D net, fairly simple. Cell traces, okay, we recorded from cell 0, and that's what that looks like. Our raster plot here. Zoom. Yeah, just some spiking, the histogram spiking pretty regularly. Ranger causalities, yeah, not very interesting. But yeah, so let's go back to edit and we'll just look real quick at what all that added. So we put in the cell types, we imported them, and we made the E and I populations. Then we could have gone in and added synaptic mechanisms. So for example, in this one, we use AMPA and GABA. And yeah, so we can see AMPA is a double exponential with the time constants. GABA is, anyway, slightly different. Connectivity rules, this time we have 2 connectivity rules because we got 2 populations. We got the E to I connectivity and the I to E connectivity. And really, you could have probably 2 more connectivity rules, E to E and I to I. So you can see how quickly you can build up more and more connectivity rules. Stimulation scores.
Just briefly, the idea here was to create like a very simple recurrent network. So that's why we connect the E to I and I to E, and that's why you get these kind of oscillations. The excitatory excites inhibitory, and then back, and you get oscillations.
Yeah, and so we put in a background stimulation into the entire population of parameter 1s. Yeah, and so on. So that, yeah, that's tutorial 2, making a model with complex morphologies.
Thank you so much, Joe. Any questions, anyone, before we move on? Feel free to speak directly and mute, or just type in the chat at any time. Okay, so we're gonna move on to tutorial 3 now. And this, I think, is one of the most interesting and exciting ones because we combine many different aspects, including a relatively more detailed network with 3 layers, like a cortical network with 3 layers. And we have the reaction diffusion involved. We're gonna calculate LFPs, and potentially we're gonna try also EEGs. And then we're gonna modify some parameters at the molecular level and see how that affects the whole network dynamics. So instead of building the network from scratch, we're just gonna load it and then look at how it, the different components look. So we're gonna go into maybe this a bit bigger, tutorials, the tutorials menu. So maybe the easiest, if you have tutorial 2 in screen, is just to go to File, New, just to make sure we clear the application and there's no issues. And just click Create here. So that clears the web application, and then we go to Tutorial. And we're gonna load this one that says 3A. Notice that there's 3A, 3B, and 3C. There are different variations of the tutorial, but we're gonna load 3A, the one that says Multiscale Network, Low IP3. Okay, so now it's loaded for me. I'm gonna leave a few seconds for everyone to catch up. The slides at this point are not as detailed, so you don't see all the steps one by one. I'm just showing here the basic steps, which is loading tutorial 3, then some of the output we're gonna see, and a minor change we're gonna make. So yeah, if you get lost or if you have any questions, please just let us know in the chat or speak up. Okay, so hopefully it's loaded for everyone. So I'm just quickly gonna go through the different components to see how it looks. So we have 2 cell types, as we saw before. In this case, if we explore the cell types, we see that they are relatively simpler. We have 5 compartments. We have 3 apical dendrites, an axon, and a basal dendrite, apart from the soma. Each of these sections has, oops, I have the section by mistake. Each of these sections has a number of mechanisms, around 10 different mechanisms, which correspond to ionic channels. So we have the passive, we have this potassium channel, we have 3 different calcium channels, another potassium channel, and this sodium channel. Okay, in terms of populations, as I mentioned, we have a laminar structure. So we have 3 layers, layer 2, layer 4, and layer 5. And each one is composed of 2 populations, an excitatory and an inhibitory. So the way we're gonna structure the network to have these layers is we're gonna specify the location or the range of spatial values that each population can occupy. And that is done in this tab here on the right-hand side called spatial distribution. So for example, for E2, if I select spatial distribution, you will see here different options, and the one that says y-axis range, this corresponds to the cortical depth. So yeah, the y-axis cortical depth, and by convention we start from the top. So 0 corresponds to the top, and the maximum would be at the bottom. So here we're saying that this layer 2 population is going to occupy the range between 50 micrometers and 150 micrometers. This is again a very simple toy model, so it's not meant to be realistic, just to illustrate these different cortical layers. If we go to a layer 4 population and spatial distribution, we can see this occupies from 150 micrometers of cortical depth to 300 micrometers. The overall size of the network, if you're wondering, is an option that's a setting that's defined by default at the beginning, but you can also change it. And it's inside this configuration panel, network attributes. Here at the top, you have several options. If you go to network attributes, you see the dimensions of the whole tissue that you're modeling. So in this case, it's 100 by 500 by 100. Again, a toy model. This is in micrometers, by the way. Okay, so if we go back to the populations, we have these 6 populations, and then the rest is similar to before. So we have these 2 different synaptic mechanisms. Here we just label them excitatory and inhibitory for simplicity. We have 2 different connectivity rules, and we're actually going to look a bit into this a bit more because they are connectivity rules that are meant to illustrate the power of NetPyNE to define connectivity. So they have some particularities. So we have one connectivity rule that's called E to all, and this means excitatory connections to all other cells, or excitatory cells to all other cells. And if we go here inside the connectivity rule and go at the top to where it says presynaptic cell conditions, we see that we're connecting on the presynaptic side all cells that are of the cell type E, excitatory. And on the postsynaptic side, we are connecting to all cells that are within the range 50 and 500 micrometers depth. This is just a way to illustrate the flexibility of NetPyNE. Notice that we can select cells for both the presynaptic or postsynaptic cells based on different attributes. And one of the attributes is the cell type, excitatory in this case, and another attribute is for example, the cortical depth. So since our network is 500 micrometers in total, this means we are connecting to all cells. So in the parameters of this connectivity, we can see that the weight of the connection follows this expression, mathematical expression. And this is something that NetPyNE allows. NetPyNE has some built-in variables that you can use. And in this case, this post-Y norm means the postsynaptic cell cortical depth. So Y is the Y coordinate, which is the cortical depth, and norm just means normalized, so between 0 and 1. So what we're doing here is saying that the weight is going to depend on the depth of the postsynaptic cell. So cells that are higher up will have a lower weight than cells that are deeper in the layers. This is an example of something that you might find in literature. In particular, we actually found that the opposite effect, where cells at the top had a stronger connectivity for a particular projection. But again, it's just an example to illustrate what you can do. And the probability of connection, we have it fixed to 0.1, and the distance, sorry, the delay between the cells. So we add a delay that represents the time it takes for the action potential to travel through the axon, release the neurotransmitter, and so on.
Can I ask a question?
Yes.
So if I have a specific connectivity rule, or some data which is fitted to the distribution of, say, photons around the axon as a function of distance from the soma, how easy is it to implement a specific synaptic function for that? So
you said the distance from
distance dependence, for example, from a cell in terms of the probability of connectivity. I'm just wondering if I have a specific function, how would I actually implement? Obviously, through the GUI, you can't do that because you've got the lack of flexibility. But is it relatively easy to plug in some specific function if you wanted to?
So even through the GUI, so I've changed to this other connection rule that we have here from I to E. And as you see here, the probability of connection is also expressed as a mathematical expression. Mm-hmm. And here we're using this distance 3D, which is I think what you were asking about, which represents the distance between the pre and postsynaptic cells in 3 dimensions. You also have a 2D if you just want to have the horizontal plane. And so, yes, so this is a mathematical expression based on this distance. You can use here a bunch of other mathematical operators that we have a list on NetPyNE. So you can define pretty complicated ones. We also can define your own constant values. Here we have like a probability length constant.
You can type into that as long as it's in the syntax, correct syntax,
yes.
A specific equation that you want.
Yes.
Okay.
But if you have a more complicated one, which is also possible, and you might have like some custom values or whatever, there's also a way to plug in your own connectivity matrix. So you can define sort of 1-by-1, 1-to-1 mapping for all of the pre and postsynaptic cells in your own matrix that you can calculate outside of NetPyNE and then just plug in if you want more customizable power. Yeah.
Okay, great, thanks.
Okay, so yeah, I was going back to this E2all connectivity rule. I was talking about the connection delay in milliseconds, which again here is a function of the distance between the 2 cells, the pre and postsynaptic cell, which is encoded in this variable, and a fixed variable that we have defined with the propagation velocity. So you can actually have a look at how to encode these mathematical expressions in the NetPyNE documentation. If you go to netpyne.org and navigate here on the site, in package reference, network parameters, functions and strings, we call it. So I'll put it here in the chat in case you're curious. Chat. There it is. So here you have basically the list of mathematical operators that you can use and functions. You have typical sine, cosine, mean, square root. You also have some random methods like binomial distribution, log normal, and so on, which are typically used for distributions. And you have a list of the built-in variables that NetPyNE provides, which are basically the XYZ location of pre and postsynaptic cells and the distance between pre and postsynaptic cells being connected. Okay, and yeah, I already mentioned for this connectivity rule, the I2E, the probability of connection here is also a mathematical expression that depends on the distance between the cells. So essentially it's saying that it's an exponentially decaying probability of connection, which is very typical of cortical circuits. And it basically means that the further away the cell is, the lower the probability or the chance of connecting these 2 cells. And this is typically done for inhibitory connections. So we'll see what the effect of this is on the network connectivity matrix we're going to plot now. Okay, so moving on, just quickly mention that this network is driven by background stimulation. Here in stimulation sources, we have created this source of stimulation that we call background. It's a net stim, which in neuron represents a spike generator. And you can define the frequency of the spike generator, which we have set at 20 Hertz, and the level of noise or randomness we have set at 0.3. We then tell NetPyNE to connect this background stimulation to both the excitatory cells and the inhibitory cells. And again, we can define different properties for this, such as where we want to connect these external inputs. We are selecting here the soma. We're selecting an excitatory cell type, an excitatory synapse, sorry, for the excitatory cells, the weight of the connection, and the delays. Notice here we're using another expression, in this case, a normal distribution with a mean of 5 and a standard deviation of 2. And we do the same for the inhibitory cells. So basically we have a background input that's driving both the excitatory and inhibitory cells. And then we have some connectivity between these 2 populations, which is a very typical setup of networks. And that's essentially it. So we're going to go ahead and create the network. Okay, so here it is. As you can see, the cells are less detailed than before. So we have these 5 compartment cells, and both the inhibitory and excitatory cells look kind of similar. So what we're gonna do is use this control panel here at the bottom, control panel, to try to visualize better the different cell types. Let's move this here. So we can color each population separately just by clicking at the network level here, this random color icon. Okay, and so now we see, we can kind of distinguish the different layers. Here at the top, we have these 2 colors, the sort of pinky and light green, which would be E2 and I2. I believe the pink ones are the excitatory cells, and these green ones, yellows, are the inhibitory cells. And the same for the other 2 layers. So layer 4 would be here, this blue and dark green. And then at the bottom, we have the dark blue cells and the sort of brownish ones. Okay. So let's look now at the connectivity matrix. Okay, and now we have quite an interesting matrix here. Collapse this. So this is showing the presynaptic populations, and how they connect to the postsynaptic populations. Notice that we have all these 6 populations, E2, I2, E4, I4, E5, I5. And with only 2 connectivity rules, we have created quite a varied connectivity matrix. So let's explore this a bit more. So for example, the connectivity between these excitatory layer 2 cells and the rest of the populations seems to increase. So this yellow, it's stronger, and here it's quite weak. Can anyone venture to explain why this is happening based on the connectivity rules that we saw before? So again, we have connectivity between this excitatory cell increases with depth.
Yeah.
Yes, sorry.
Yeah, that's what I was going to say. Increases with depth.
Okay. Great. Fantastic. So yeah, so that's essentially, we can go back now and look at it, but that's the mathematical expression that we have. If you remember, was a function or was proportional to the depth of the cortical cells. So we see here that that's what's happening here. The deeper ones in layer 5 get a much stronger weight. That's what we're seeing. And similarly, we can have a look at, for example, the inhibitory cells. And this layer 2 inhibitory cell, and the weight connects to other cells in the same layer, in particular to other excitatory cells, seems to change based on the layer. So here, I2 connects very strongly to E2, but very weakly to E4 and E5. Whereas, for example, E5 has the opposite pattern. So I5 has the opposite pattern, so very strong to the layer 5 and very weak to the other layers. Can anyone explain this based on what we saw before? So again, the connectivity of the inhibitory cells seems to connect very strongly to the cells in its own layer, to the excitatory cells in its own layer. So I2 very strongly to E2, and E5, for example, very strongly to E5. Sorry, I5 to E5. Okay, so this is basically because we had this distance-dependent connectivity where the probability of connection decayed exponentially with the distance. So that's why I2 connects very strongly to its own layer. And here in I4, for example, you're seeing the effect of these being probabilistic connections, where you would expect a much higher probability of connection here in E4, but due to probability, it's actually only slightly higher. than E5, for example. And yeah, and for I5, you see the effect very clearly. So it's very strong for layer 5 and weak for the others. Okay, so let's move on and let's now simulate this network. And while this is simulating, let me explain. I forgot to mention before that this network has reaction diffusion, so we didn't actually look at the reaction diffusion parameters. But let me explain how the reaction diffusion mechanism works. So what we have here is the excitatory cells of this network. They have several components, particularly this endoplasmic reticulum, which is this internal organelle that has some this CERCA pump, these IP3 receptors, and a bunch of processes here. So I'll go step by step showing what this is trying to simulate. So basically, we're trying to simulate the effects of metabotropic glutamate receptor, which is shown here in number 1. And what this does is when the metabotropic receptor activates, it increases the level of IP3. IP3 is just a molecule that has some effects in this cell. And so it increases the level of IP3 inside the cell, inside the cytosol. The cytosol is what we call the inner part of the neuron, of the cell, the soma. So once this IP3 increases in the cytosol, it binds to these IP3 receptors in the endoplasmic reticulum, and these IP3 receptors open and release calcium. So they release calcium again to the cytosol, and so the level of calcium increases. This is number 4. As the level of calcium increases, these calcium molecules bind, attached to the calcium-dependent potassium channels, which are shown here in 5, which are channels that basically activate or open when calcium binds to them. And so by these potassium channels opening, they allow potassium to flow into the cell. And this, of course, hyperpolarizes the cell. You get a hyperpolarizing potassium current and overall reduces the firing of the neuron. Okay, so we're seeing here a multi-step chain reaction involving different molecules, IP3, that binds onto IP3 receptors, opens the ER channels, releases calcium. Calcium binds to calcium-dependent potassium channels, and this opens potassium, which reduces the firing of the cell. All this is modeled in this model that we're just running right now. Okay, and it has finished the simulation now. So let's explore some of the plots. So in the cell traces, I think we can see a bunch of things. So we have recorded the voltage in the soma of this excitatory neuron. So we're getting some spikes here. We're also recording the intracellular concentration of calcium. Okay, which we see increases steadily over time. And the extracellular concentration of calcium, which drops because this is being used by the channels. And we see these potassium currents, which are the result of these potassium channels that we have along the axis, along the membrane. Okay. So we can also look at the raster plot. So we see all the cells in the different layers are spiking. We have these sort of oscillations, and the network is relatively synchronized. The spiking histogram shows a similar effect with these oscillations over time of the different populations. We also have the local field potential. So what we did here in this network, it's set up, let me put it here, to record, there are 3 electrodes, and the average is at the top. So we here, the white one is the average, and then we have 3 electrodes at different depths. So they were placed at the 3 different layers. And so we're seeing the LFP recorded from layer 2, layer 4, and layer 5. We can actually also visualize the power spectral density to see the peak oscillation frequencies of each of the electrodes. And we see the average is around 24 hertz. The top layer has these very strong high frequency oscillations. Layer 4 shows the peak at 25 hertz, and layer 5 shows peak at 26 hertz. And this plot here shows where we actually recorded or placed the electrodes. Let me see. Oh no, sorry, this is the spectrogram. So similar to the power spectral density, we can see, let me make this bigger. We can see the average shows this oscillation frequency over time around 20, 25 hertz. It's a bit more diffuse in the top electrode, where you have this very high gamma power. And then the layer 4 and layer 5 show these oscillations at around 25 hertz. Okay, and finally, the other plot I want to show here is this RxD concentration plot. So this one is interesting because it's showing the concentration of calcium at the extracellular space. Okay, so we see this is the volume of the size of the column, which is 500 micrometers depth by 100 or 200, 100 micrometers horizontal distance. And the concentration of calcium is fixed along all the extracellular space, except for the locations where there are neurons. And this is just a slice across. We're catching only a few neurons. And notice where there are neurons, you see this decrease in the level of in the concentration of calcium as calcium is being used by the neuron. So we have so there's a question in the chat: what is Granger causality? This is an information transfer measure. It basically tells you. Let me see if we can get something here. It tells you how much of one population can be predicted by the activity of another one. So it's a kind of a measure of causality, but also more of correlation, really. And you can select different populations to look at. Here, see, for example, that the causality from E2 to I2 is quite high at a certain frequency, meaning that in theory, E2 is causing the activity of I2 at that frequency of oscillation. Okay, so yeah, just to highlight here that we're looking at information at different scales. And on the one side, we are seeing here the concentration of calcium in the extracellular space, but we also have access to, for example, these LFP recordings from different layers. So any questions before I move on to make some changes in this network? Okay, so we're gonna go back to the edit mode here, and we're gonna make a change in the reaction diffusion parameters. So we're gonna make a change at the molecular level, and what we're gonna do is, well, let me go over how this reaction diffusion is organized. So this follows the same structure provided by the neuron simulator, which basically has these different components for reaction diffusion, regions, which defines different regions within the cell and outside of the cell, like the extracellular space. We have species, which are different molecules that are moving around. In this case, we have calcium and we have IP3. And for regions, we have the cytosol, we have the endoplasmic reticulum, we have the membrane between the cytosol and the ER, which has some special particularities, and so it's defined separately. and we have the extracellular space. Okay. We have the species, and then we have states, parameters, reactions, multi-compartment reactions, and rates. So I'm not going to go into the details of all of these. There are very nice tutorials for RxD provided online on the website, and you can see different examples. But just to mention, for example, here, this multi-compartment reaction, where we have 3 of them. One of them is this Cerca pump in the endoplasmic reticulum. And then we have, for example, these IP3 receptors, where the IP3 is binding to these receptors, and then that's releasing calcium into the cytosol. Okay, so what we're going to modify here, just to see the effect it has on the network, is a property of one of the species. So this IP3. So if you remember, IP3 was the one that increased initially and then started the whole reaction, leading to a decrease in the cell firing rate. So let me go back here. So if you see here for IP3, we have different properties, and one of them states the initial value. So the initial value is set to 0, so there's no IP3 initially. So we're going to change this to 0.1. So we're going to say basically that there's more IP3 initially than in the original model. Okay, so everyone please make this change. So you have to go to reaction diffusion, select species, select IP3, and where it says initial, change from 0 to 0.1. You also have this on the slides right here. Let me know if there's any questions or issues. Okay, and once we have made this change, we can simulate the network again. Notice that here we can just go to create and simulate and click here to do everything at once. So based on this reaction here that I've shown, can anyone guess what's going to happen in the network? What's going to happen to the activity if we increase the initial concentration of IP3?
It'll have reduced activity.
Okay, great. So let's see if that's the case. So this model in particular takes a bit longer because, well, it has a few more cells. I think there's 16 total with 5 compartments each, but also particularly because it has the reaction diffusion in all excitatory cells. And of course, reaction diffusion takes quite a while to simulate. Okay, here we go. So let's look, for example, at the raster plot. Okay, and here we already see some changes. So after around 500 milliseconds, we start seeing a decay of activity that wasn't present before. We can also see this very clearly in the local field potential. So we see after 600 milliseconds, especially, the power of the LFP, the amplitude goes down quite drastically. This, of course, will change slightly the peak frequencies. And we can also have a look at the voltage, the traces that we record. And yeah, so for example, in this excitatory cell, you see over time the membrane voltage with the spikes decreases, and you stop having spikes. You see a much faster increase of the level of intracellular calcium because this has been released by the increased level of IP3. So notice that it takes some time for the IP3 to take effect because you have all the processes, the molecular level processes happening. But after some time, you get a quite fast increase of intracellular calcium. That's the one that's opening the calcium channel, the potassium channels, and reducing the overall activity. So you see here this increased potassium currents, and then the network sort of dies off. Okay, so yeah, we have, just to summarize, we have basically seen this 3-layer cortical network with reaction diffusion, and we have modified a molecular level parameter, which is the concentration of a molecule, IP3, and that has resulted in changes that affect the spiking of the single cells and in turn of the whole network activity, including the network activity recorded by these local field potentials. Any questions, comments? Does it have a way to plot before, after modification plot? I guess you mean visualizing both at the same time, and there's currently no way to see both at the same time. Of course, you can save figures. All of these figures that are generated here, you can save them, and you get a PNG file. And so, of course, you can save the previous one and this one, and then compare them on your local machine. Okay, Angus, thanks. Any other questions? Okay, so we can move on to the final tutorial for this session, which is going to be parameter exploration using the GUI. And this is quite exciting for us because it's the first time we actually showcase this in an online tutorial, because the feature has been recently added. So let's see how it goes. Again, as before, the first thing we're going to do is just clear this. Actually, I think we can directly load the new tutorial, and that clears automatically the previous one. So let's just go to tutorials, simple tutorial 4, simple oscillatory network. This is the one we're going to use for the parameter exploration, because it's quite a small one and you can run simulations pretty fast. So yeah, let's everyone click here, tutorials, tutorial 4. We confirm. Hasn't seemed to clear the whole plotting stuff, but I think if we go back to edit, yeah, we should see the new model. Okay, so yeah, I hope this worked for everyone, and everyone is now on tutorial 4. And so we're going to do the same. We're going to have a quick look at the different components of the model. We're going to run a single simulation just to see how it looks. And then we're going to create these parameter explorations. So this is a very simple model. We just have one cell type, which is a pyramidal cell type with a single compartment, a soma. So we want this to be very quick. we have two populations, an excitatory and an inhibitory, but note that because this is a very simple model, both are made up of pyramidal cells. This doesn't make any sense, but it's just, again, a toy example in this case. We have two types of synapses, the excitatory and inhibitory, and we have these two connectivity rules, which basically make the network oscillate. So this is connections from the excitatory cell to inhibitory cells, and from the inhibitory population to excitatory population. When you have this kind of recurrent connectivity, it's very easy to get oscillations, which is what we want to demonstrate in this case. To start the network firing, we have this stimulation source, this NetStim, a spike generator that essentially targets all pyramidal cells. So in this case, all the cells in the network. We don't have any reaction diffusion. And yeah, we can just go ahead and create and simulate this. Actually, before you create and simulate, we're gonna make a small change. And I wanna illustrate the EEG plotting with this example. So what we're gonna do is go to the configuration panel. Okay. And inside the configuration panel, we're gonna go to record. So notice here, it's all the things we want to record from this network. You can define different cell traces like the voltage or different currents. And you can also specify recording of these dipoles. So dipoles, as I mentioned before, are the current dipole moments, is the more strict name. And this is what is required to calculate the EEG signal. It's basically the total amount of current coming in and out of the cell at each location of the cell. So we are going to select these record dipoles using LFPyKit method. LFPyKit is another package that allows you to record and make these plots very easily. So NetPyNE makes use of this package. And so once we click this, it's going to be automatically saved for all the cells, and then we can plot EEG for the network. So again, quickly go to the configuration panel, record section, and select record dipoles using LFPyKit method. I'm not sure if I added this to the slides. Let me check. No, I don't think so. Let me do that quickly, just in case somebody gets lost. I'm gonna take a quick screenshot here. Okay. Record current dipoles to plot EEG. Okay, perfect. So let's create and simulate the network. Okay, so we see the network now is very simple. All the cells are just composed of a soma. So this is very quick to run. We can color the two different populations separately. So we see that we have excitatory and inhibitory cells. And then let's just look at the activity. So we can look at some voltage traces of one of the cells, which is very clear, nice spikes. And we see the activity of the network with the two populations, the excitatory at the top, at the bottom, and inhibitory at the top. And we see that we are already getting some kind of oscillation. So the network is tuned. This is more clear here in the histogram. Yeah. So we have these already nice oscillations that you can see. Okay. So what we can do now is try to plot these two new plots here at the bottom. Oh, well, and just notice that the oscillations are around, I think it says here, 14.5 hertz is the average rate. But the oscillation, you can sort of count the peaks that you get here, and you will know the oscillation frequency of the network. So it's around 20 something, I believe. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 18, 19, 20, 21, 22, 23, 24. Yeah, around 24 hertz. Okay. So the two new plots that we have here are the dipole plot and the EEG plot. The dipole is just going to show the average dipole moment of each cell summed together, and which is a precursor of the EEG plot. Ooh, okay, so this one is not working, and this points to an issue with memory. So I'm just gonna try the EEG plot, but I think it's gonna have the same issue because we're all logged in at the same time. These plots require a lot of memory, and so it's not clear that they will always work with many users at the same time. And yeah, so the EEG plot didn't work here. Okay, so unfortunately, we're not gonna be able to see the plots, but yeah, I can show you briefly how they look if I have. Actually, we'll see them in the session 2 this afternoon. We're also gonna play with those, and hopefully those will be working. But otherwise, I can show them as saved files. No worries here. We're just gonna go back to the idea of this tutorial, which is focused on parameter exploration. So just remember that right now we have a frequency of around 24 hertz. Okay, so what we're gonna do now is create our first experiment. And experiments are just simulations where you choose one parameter and change the values of it. To do that, we go to Experiment Manager, which is here at in the left-hand side menu at the bottom, Experiment Manager, and you will see this icon to create new experiment. Let me see if this is in the slides. Yes, so here it mentions we have to go to the Experiment Manager. Okay. So if we click on Create New Experiment, confirm, and now we have this new view where we are creating an experiment. Let me know if anyone is having issues with this. Okay, so what we're gonna do first is give this experiment a name. And here I'm just gonna call it oscillation, for example, OS, since we are gonna aim to change the oscillation of the network. And below that, we see here we have a list of parameters that we wanna explore. So this has a nice interface where you basically have access to all the parameters of the network. And they are labeled and arranged based on the NetPyNE structure, which we will see more of this afternoon. And so the parameter that we want to change here just to try is the time constant, the decay time constant of one of these synapses, the inhibitory synapse in particular. So it is currently 5 milliseconds. We want to try different values for this synaptic decay constant. So this is a parameter that specifies sort of the time period that the inhibitory synapse is going to be active for. So let's look for this parameter. If we type sin, we already find it here. So it's NetParams, which is the overall structure that contains all the parameters. Inside, we have the sin mech params, synaptic mechanism params. Inside that, we have the inhibitory synapse. And then inside, we have this tau2, which is the parameter we're interested in. So we can just click on that. And I'll let you a few seconds if you want to try that on your own. So again, to sum up, we go to the Experiment Manager, create new experiment, give it a title, and then select this parameter. Okay, and now we just have to tell NetPyNE what values we want to explore within this parameter. So what values do we want to try? And this again is a very typical setup for exploring models. You select a particular parameter and you change the value to see what effect it has on the network. So there are two ways of specifying values. You can use this range where you specify the initial value, the final value, and the step. So I could say something like from 0 to 30 in steps of 5, for example. Okay, so that would go 0, 5, 10, 15, 20, 25. Okay. Or you can select a list of values. So you go here, you click and select list, and now you type the specific values one by one. So this is what we're gonna choose, and I'm gonna select 3 different values. 0.1, you have to separate the values with commas, 5 and 30. Okay, Warwick. Warwick, did you have any comments or?
Hello?
Hello?
Hi. No, sorry, I was just dropping back in.
Okay, sorry.
Sorry, other bits. All going well, I hope?
Yes, all good, yes.
Okay. There's some issues with GitHub, so I think if there's any interaction with GitHub pulling things down, it might be problematic, but.
Not yet, this afternoon we will have.
Cool.
Okay, so we have defined this new experiment where we're gonna explore the effect of different values of this inhibitory synapse tau2 parameter. And so we are gonna go ahead and create the experiment. We have selected 3 values, 0.5, 5, and 30 milliseconds. And we now click create. So when we look now at the experiment manager, this is gonna have our new experiment here defined. The title is OS for oscillation. We have the date when it was created, and we have the stage it is in right now. It says design because we are currently sort of defining it. We can still go to edit here, change values, and then save. Once we are happy with the experiment defined, we can go ahead and simulate it. So now if we click on the create and simulate or simulate, we're gonna get a different screen, which asks you whether you wanna simulate just the base model, so the same thing we simulated before with the default parameters, or where you actually wanna simulate all the conditions in your experiment. So notice that we have 3 conditions because we set 3 parameter values, 0.1, 5, and 30. Okay, so we're gonna select these all conditions, 3, and click on simulate. I'll let a few seconds for people to check the screen and do it on their own. So again, after creating the experiment, I just went to the top, create or create and simulate, click on that on the rocket. Then you get this screen and you select all conditions. And we now click on simulate. And it tells you the experiment started. You can view the experiment status in the experiment manager. Okay, confirm. And we see now the experiment called OS has changed from design to simulating. This means that NetPyNE in the background is running 3 different simulations, and we'll let you know when they're completed and we can explore the results of those simulations. Notice that because this is happening in the background, you're still free to move around, look at your network parameters and so on. And when you want to check, you can go back to experiment manager and see how it's doing. So for now, it's still simulating. Again, let me know if you are having any issues. Okay, so since we're all going to simulate this at the same time, of course, there might be some more delay to complete the simulation, but it should get there eventually. So you can also click on top of the experiment and you will get more details. So this is the experiment oscillation inside, and we see the 3 different experimental conditions where we changed a single parameter value and we gave it 3 different values. Of course, in the experiments, you can always define multidimensional experiments where you select 2, 3, or more parameters, and then you will get the combinations, the different combinations of all those parameter values. And you have to be careful because it can very easily grow exponentially. We have many parameters with many values. Okay, so now we see here that it actually completed, it's finished, and it says simulated. So now we can click on this experiment, which has completed. By the way, just remember, we are recording the whole tutorial and we will post this on YouTube as soon as possible. So if you've missed anything, I will share the link to YouTube so you can follow this again on your own time. Okay, so we see these 3 experimental conditions for the different parameter values, and we can now click on this icon here, explore results, to visualize the results of each of these simulations. We can close this 3D representation, and we can open plots for this particular simulation. Okay, so we see here the raster plot has changed. If we remember before, the original value was 5 milliseconds, and we had here the average rate was 14 Hertz. So when you change the inhibitory synapse tau2 to 0.1, you get a higher firing rate. We can also see this in the spike histogram, where obviously there's less synchrony here, there's more activity and higher overall oscillations in the network. Okay. And if we now open the results for the 3rd condition, the 30 millisecond condition, I click here, confirm. And again, we open the raster plot, for example, we see a drastically different result where the spiking of the spiking in the network has basically died off. So this 30 millisecond synaptic time constant was too much for the network. Basically, all the excited results are getting inhibited too much. And if we look at the spike histogram, we see that the oscillations in the network have died off as well. Okay, so this is quite intuitive and easy to play around with. So we have these 3 conditions with different values, and we can load the results of each of those and visualize them one by one. And you cannot actually see them at the same time, but it's easy to change between one another. So this is the 30 millisecond case. I can just click here and look at the 5 millisecond one, which is clearly different and has these oscillations. And finally, just before we go, I want to show the other components here of the experiment condition. So we can click on this view model, and this is going to show you a JSON viewer, which basically contains all the parameters of this condition. So this is all the sort of the NetPyNE structure containing all the different parameters of the model. And you can see, for example, in the synapses down here, synaptic mechanism params, the inhibitory, the tau2 here was 0.1. Okay, so this is one of the value that we modified. So if you go back and open condition 3 and look at the model, you will see that the value is 30 instead. The tau2 is 30. Okay. And finally, what you can also do here is if you decide that one of these conditions is what you were looking for and you want to play around more with this model, then you can select here load model specification, and this will replace your current model definition with that particular condition. So now in theory, if we go to synaptic mechanisms and we check the inhibitory synapse, yes, so we see here the time constant is 0.1 instead of the original 5 milliseconds that we had. Okay, so I think that concludes this parameter exploration tutorial. Please let me know if you have any questions or comments before we go for a break. Okay, so thanks everyone for attending this morning session. We're gonna break now for lunch break or whatever you are, might be a different thing, but we're basically coming back at 12:30. So we now have around 45 minutes of break. Let me show the schedule again, which is right here. Okay, so we finished this session and now we have this 30-minute, which is now 40-minute break, and we'll come back at 12:30 ET time, Eastern time, for the second session where we're gonna see actually quite similar things, but done programmatically via scripting and Jupyter notebooks. Thank you guys. And we're also gonna look at importing cells using these scripts and how you can run and take advantage of the Jupyter Lab in open source Brain to build models and explore some of the models we've already built. So thank you so much and see you in the afternoon session.